So in this video we're going to think about gene cloning. We already know that cloning is all about making identical copies. So what do we need to know in order to be able to do gene cloning? So we need some scientific knowledge. And we already know about the structure of DNA. The fact that it's a double helix, it's got bases, and essentially it is universal in all living things. So DNA in bacteria and DNA in plants and DNA in animals, it works in all of them. So if we want to do transcription and translation, we have a universal code. So we can switch out DNA from plants and animals and into other organisms and quite happily still get proteins made. The other things we need to know about, we need to remember about enzymes. If we're going to be moving DNA around, we're going to have to cut it out of one organism and put it into another one. So you have to know how enzymes work and how their active site will recognise specific parts of the DNA that we want it to recognise. We're also going to need to remind ourselves of the differences between prokaryotes and eukaryotes in terms of how they structure their genes, and we'll need to make sure that the regulatory parts of our genes are included if we're going to move DNA from a eukaryote to a prokaryote or vice versa. So now we know what scientific knowledge we need, let's see what other aspects we can think about with gene cloning. We have already met gene cloning in a test tube, and this is called PCR, or polymerase chain reaction. And this is a series of events that happen in the laboratory under controlled conditions, where we take a double-stranded piece of DNA, perhaps with our gene of interest, maybe from a crime site or maybe from COVID testing, and we go through a series of reactions. We start off by heating it up to 95 degrees centigrade. This causes the DNA to fall apart. Then we use a primer to attach to the DNA, and we use an enzyme which doesn't mind being at a hot temperature. It's a special enzyme called Taq polymerase. It comes from a bacteria that lives in very hot springs, and that Taq polymerase can take those single-stranded pieces of DNA and join a new piece of DNA to it, using that first one as a template. So you've gone from having 2 pieces of DNA, now you have 4, and you just keep going through rounds and rounds of PCR and cloning, making new copies of our gene of interest. So that's one way that we can do gene cloning. We'd like to do it in living organisms. So in living organisms, we are now going to make what are called recombinant DNA structures. So recombinant DNA, because we are recombining DNA from different organisms. That means we're going to end up with a transgenic organism. In other words, an organism that has genes from other areas. So in a bacteria, you could put a gene from a plant or an animal, and you now have a transgenic organism. In the popular media, we call this a genetically modified organism. Or GMO, and sometimes we call this genetically engineered. Genetic engineering, or a genetically engineered organism. So different terms, all for the same thing. So let's see how we do it. So we need to find our organism that's going to do the hard work for us. And usually we use something simple that's easy to grow in the lab, maybe like a bacterium. So remembering that our bacteria has two sorts of DNA. The chromosomal DNA, we're not really interested in that, that's a big loop. We're interested in the tiny loops, the plasmids. And the plasmid can be taken out of the bacterium, and if we blow it up so we can see it a little bit more detail, we can make a cut in that circle of DNA with a restriction enzyme. You can call restriction enzyme RE for short. And now we don't have a circle, we have an open circle. And into that open circle, we can place some DNA of interest. So where do we get that from? We get it from, maybe from us. So we've got our chromosome, and we take a small portion of that and cut it with a restriction enzyme, and we end up with our gene of interest. This is the gene that we want to clone and make more of. So we take this gene of interest that's been cut with the same restriction enzyme, and we bring it in the laboratory into contact with our plasmid that's been cut open, and we combine these two together so that we end up with a circle that is now slightly bigger than it was before because it's got our gene of interest inserted in it. And it's held in place by using another enzyme called DNA ligase. So it joins the two pieces of DNA together. Now, this circle we can now place into another bacteria to grow it up for us. So we put it into a new bacterium. That's a process called transformation. We've got to transform this new bacteria and give it our little plasmid with our gene of interest in it. And we now use our knowledge of how to grow this bacteria, giving it perfect conditions of temperature, food, oxygenation, and this bacteria will undergo binary fission and do transcription translation for us to make lots of our protein from our gene of interest. This is using recombinant DNA technology. We've recombined DNA from two organisms inside a genetically modified transgenic organism. And that's gene cloning.